necessary to speak or can I speak Arabic also?
Again, please.
Is it necessary to speak English language or it's possible to speak Arabic?
No, as you like.
Okay, thank you so
No,
much.
English mostly. And if you want some Arabic, okay, but mostly English, okay? Please.
Okay, okay. Okay, so I will start now. Assalamu alaikum. Welcome all of you. My presentation today will be under the title of DNA identification in mass disasters, missing persons, and forensic investigation. I'm Mabroka Alghros, the technical advisor in the National Authority for DNA Fingerprinting Research and Analysis in Libya. So as I mentioned, I'm from the National Authority for DNA Fingerprinting Research and Analysis. I'm here in cooperation with the Genetic Jordan and the event under the supervision of Palestinian Association of Biotechnology. So when we are talking about a human identification in general, we have to face 4 phases for this process, because when we find the crime scene or the mass disaster scene, we have first of all to make a scene examination, to collect the data, to see how many human remains, how many bodies in that scene or in that position. The other stage is post-mortem or BM data. It's depending on the forensic pathologist to describe the body, the cause of death, and if there is a special marks in that body or that human remains. After that, we will start to collect the antemortem data or AM data from the family, the relatives of the missing person or the victim of the armed conflict or disaster in that area. The phase number 4 is reconciliation, or to collect all the information together and compare it. The AM data from the family and the BM data from the scene and from the forensic pathologist. And after DNA analysis, in the traditional schools, when we use just the appearance of the body and the age, the sex, the structure, the height of the individual, they compare the information, and after that they go directly to the matching report if that possible. So we have to mention again that DNA profiling is the gold standard for human identification. This technique has been widely used successfully, not only in forensic and crime investigations, but also in the identification of victims in mass disasters and missing persons. في المجمل عملية التعرف على الهوية في حالات الكوارث الطبيعية أو في حالات النزاعات المسلحة وأيضا في قضايا المفقودين تعتمد خلال السنوات الأخيرة بشكل رئيسي وجوهري على تحاليل البصمة الوراثية أو ملف الحمض النووي ومقارنة هذه البيانات مع العينات المرجعية التي يتم جمعها من أهالي المفقودين أو من أهالي الضحايا. So the key biotechnologies that may facilitate the identification process, such as DNA extraction, real-time PCR for quantitation of DNA, and genetic analysis for short tandem repeats, many STRs, and single nucleotide polymorphism. And we also use some software and computer programs which are employed to analyze biological data to facilitate the matching of remains with reference samples, enhancing the accuracy and efficiency of identification process. So if we are talking about the application of biotechnological technique used in DNA profiling, you have to mention many fields, including solving crime, murder or sexual assault, missing person, and unidentified remains. So also for establishing biological relationship, child support and paternity test, and also to detect the identical, if they are twins, identical twins or no. We can say also identifying victims of man-made disaster, not natural disaster, and also identifying victims of natural disasters. So when we are talking about a human identification process, many specialists work together in this field. Forensic pathologist and forensic dentist, forensic anthropologist, and also the technician in the lab. When we are talking about forensic pathologist, they perform autopsy and they detect the cause of death. Also, they collect the biological sample, they collect the information, and also the forensic dentist who compare the dental records from the human remains with the data, AM data, or antemortem data from the missing person or the victim in this event. So identification methods include examining skeletal features, dental record, fingerprint, distinguishing marks like tattoo or scars, medical devices, personal effect, and finally the DNA profiles. When we are talking about the forensic anthropologist, the anthropologist is a specialist for the bone, because some victims, in some cases, the human body decompose and only the bone, the human remains will be only bone. So the anthropologist will play an effective role in this case. So he will detect if the remains are human or not. He will determine the sex of the human remains, the age, the estimated age, the ethnic origin, and the height. All this information is provided by the forensic anthropologist. After collecting the data and complete the work from the forensic pathologist and the forensic anthropologist, the sample will transfer directly to the laboratory, forensic laboratory. So the main work in the lab, starting from sample receiving, storage and preparation, DNA extraction, DNA quantitation to detect the quantity of DNA in the sample. Is it enough to go forward to end the analysis or no? After that, we need the step of PCR amplification, genotyping to detect if we are working on fragment analysis, STR or single nucleotide polymorphism. After getting the raw data from the genetic analyzer, we will go directly to data interpretation in special software. And we will finally issuing the matching report to mention if we find match for this human remains with the reference sample from the families. So when we are starting in the lab, the first step is preparing the sample, preparing the bone sample. So we have to sand it to remove the outer layer from the bone. After that, we will wash the sample by chemicals. And finally, we will grind the sample to convert it to fine powder, because the fine powder is the target to make the extraction of DNA from the sample more successful. So we can grind the sample either manually or by instrument. If we are working manually, we can use the manual way directly, or use the drill to make a hole in the sample and collect the powder from this process. We also have tissueizer, which work directly by disrupt multiple biological samples through high-speed shaking in tube or 90 or 96 well plate with stainless steel. Or we can go directly to use the freezer mill instrument, which using the liquid nitrogen, which will be minus 100 and I think minus 100 degree. So we will keep the sample very cold, and the heat will not affect the sample, because the heat is one of the real cause of sample degradation or DNA degradation. So we use the liquid nitrogen in this instrument to be sure that the sample during the grinding is still stable and not affected by the heat. So after converting the sample to powder, we will go directly to extract the DNA from this powder. We use automated extraction instruments, either from QIAGEN or from Applied Biosystem company, which working by the magnetic beads extraction way. So after we get the extract of DNA, we will go directly to the quantification of DNA. The quantification in this step, we need to know if the sample is it enough to go further in the analysis process, and also to detect if the sample is human DNA or no. Is it male or female? This is depending on the kit used with this instrument, genetic qPCR from Applied Biosystem. Okay, so in this stage, we will quantify the DNA to be sure from the quality and the quantity of DNA. If anything was bad or was wrong, we will have to go back to the extraction step or the preparation and grinding of the sample. So after the quantification, we will be sure that we have the enough amount. Maybe it will be concentrated, so it need more dilution for the DNA sample, or it will be not concentrated or little amount, so we have to concentrate the amount to go to the next step. So in the PCR amplification, we will amplify the target fragments or sequence, adding fluorescent label primers to visualize the target fragment in the genetic analysis step by genetic analyzer. So the next step is the fragment analysis of the genetic analyzer. The genetic analyzer is an advanced instrument used in forensic laboratories for DNA analysis. It performs high throughput sequencing and genotyping, allowing for accurate profiling of DNA samples, including short tandem repeats, commonly used in human identification. In a forensic DNA human identification lab, the genetic analyzer plays a crucial role in analyzing DNA from crime scene, missing persons, or disaster victims. It helps produce DNA profile that can be compared against databases to identify individuals. So when we are talking about the last step in genetic analyzer, we will have the raw data. The raw data then will transfer to another software to make raw data interpretation. We have more than one software. It's depending on the software available in the lab. We have GMapper IDX from Applied Biosystem. We have Osiris from NCBI, and we also have GeneMarker. All of them working on interpretation, the raw data from genetic analyzer, and converted to visualized result, visualized DNA profile to go further to match these profiles with the database of reference sample. So this is the typical form of DNA profile. It's consists of more than panel with different colors, and also it's composite of more than one locus. So once we get the DNA profile from the sample, which comes from the human remains and identified body, we will go directly to the matching software, especially in mass fatality, when the number of victims are huge, are big. The manual work, it's okay, but will not be enough or professional in this stage. We have to work on matching software. One of the most famous or most valuable, its emphasis, mass fatality identification system software. There is also GeneMarker HID, and there is another one, it's open source, called Familias. So you can use one of these software to compare the sample and to get the match of the sample with the reference samples. So in this table, we have a list of software programs for paternity testing and kinship analysis, and the final decision for the lab manager to choose the best or the appropriate software for its lab. When we are talking about the typical situation in human identification by DNA, we go directly to the short tandem repeats to the nuclear DNA. But in some circumstances, the amount of DNA couldn't be present or couldn't be enough to make this STR analysis. And sometimes the human remains are very old, ancient, so the nuclear DNA are degraded by the time. So we go to the mitochondrial DNA. Mitochondrial DNA play a vital role in human identification due to its high copy number in cells and maternal inheritance pattern. It's particularly useful in case where nuclear DNA is degraded, limited, or not available, such as in ancient remains or hair or bone. We prefer mitochondrial DNA in situations where degraded sample, mitochondrial DNA, is more resilient than nuclear DNA, making it ideal for analyzing old or damaged sample. Relative of missing person, since mitochondrial DNA is inherited maternally, it can be used to identify individual by comparing it to maternal relatives, aiding in missing person cases. Forensic analysis, mitochondrial DNA help in identify victims in mass disaster or when nuclear DNA is compromised, enhancing the accuracy of human identification effort. Overall, mitochondrial DNA is a valuable tool in forensic science and anthropology for its unique properties and applicability in challenging circumstances. The same situation, if we don't have all the family, or we couldn't find the close relatives of the missing person or the victim of a disaster, we can go directly to the Y chromosome STR, which work on the paternal lineage, so it can connect the victim to its relatives from the paternal side. When we are talking about the advances in forensic identification, we can talk about forensic DNA phenotyping. It's a technique that predicts physical trait and ancestry from DNA sample, particularly useful when traditional DNA profiling is not viable. In some circumstances, we couldn't find enough sample from the crime scene or from the missing person or from the victims. So we can directly go to the single nucleotide polymorphism. And the literature review, the previous study about this topic, give us a clue to predict the eye color, the hair color, the skin color. All of these could give us an image about the phenotype of the person. So it involves analyzing single nucleotide polymorphism, which are variations in a single nucleotide in the genome. Commonly analyzed SNPs in forensic phenotyping include those associated with, as we mentioned, with the hair color, and is the number of single nucleotide polymorphism, and eye color, and also with skin tone or skin color. Okay. In such circumstances, we are talking about armed conflict, about natural disaster, about missing persons, we have to talk about DNA database. DNA database could be national DNA database, or could be international DNA database, or forensic DNA database. In certain countries, let us talk about disaster, natural disaster happen in some country. The first step is, after collection the DNA profiles from the relatives or from the human remains, we have to go directly to create national database. The national database primarily uses it to solve criminal cases, but also for identifying missing person. For example, the codes in the United States include a missing person index to help in identification. When we are talking about international DNA databases, which use it for cross-border cooperation in identifying disaster victims or missing persons internationally. For example, we have the Interpol DNA Gateway. When we are talking forensic DNA databases, it must or it could be part of the national database, or it depends on the country policy. So forensic DNA databases, it's used in criminal investigations and contain profiles of known offenders and crime scene evidence. As an example for a forensic DNA database, the European Forensic Database. So there's another aspect from DNA profiling and human identification and databases. It's the data privacy in DNA technology and the human identification. Data privacy is very, very important and crucial aspect. So first of all, we have to work on protection of personal information because DNA contain highly sensitive information, including genetic trait, ancestry, and health risk. Ensuring the confidentiality of personal data is critical in forensic investigation and the human identification. Legal and ethical guidelines. National laws and international agreements regulate how DNA data can be collected, stored, and shared. Forensic labs must adhere to data protection laws such as GDPR in Europe. Informed consent. Individuals should provide informed consent for their DNA to be used in identification cases. In mass fatality cases, consent from family members may be required for comparative DNA analysis. When we are talking about data privacy, we have to mention data retention and deletion. Once a case is resolved, DNA data should be probably archived or deleted based on legal guidelines. This prevents misuse or unauthorized access to personal genetic data. When we are talking about challenges in international databases, we have to mention that cross-border investigations can complicate data privacy due to varying legal frameworks. Collaboration between countries must ensure privacy protection while allowing effective DNA matching. Technological security measures, databases should be encrypted and secured against breaches. Access to sensitive DNA data should be restricted to authorized. The conclusion of this presentation that DNA technology plays a crucial role in mass disaster identification and forensic investigation, and advances in biotechnology, such as forensic DNA phenotyping, mitochondrial DNA, and Y chromosome analysis, allow for more accurate and efficient identification. Data privacy and ethical consideration are paramount in ensuring the responsible use of DNA in a human identification. Thank you for your attention, and I'm open for any question. Do you hear me? Hello?
شكرا لك أستاذة مبروكة. هلا بننتظر إذا في حدا عنده سؤال أو استفسار. Any questions for Dr.
So it seems there's no questions. Everything is clear. So let's see. Okay. So thank you. This presentation will be our last English presentation. We still have 3 presentations in Arabic. The next one will be at 4:25 p.m. That means after 20 minutes. So
Okay. if I still have a time, if you don't mind, I can speak Arabic to the audience who speak Arabic.
Okay, no problem. If you have anything to say, you can say it.
Okay, thank you so much.
You're welcome.
اوكي السلام عليكم مجددا. العمل في مجال التعرف على الهوية باستخدام تقنية الحمض النووي هو عملية حيوية جدا، خاصة في ظل الظروف التي تمر بها دولنا، ابتداء من ليبيا وصولا إلى دولة فلسطين. في هذه الظروف، مع وجود عدد هائل من الوفيات وعدد هائل من البقايا البشرية التي لا يمكن تحديد هويتها عندما تصل إلى مرحلة من التحلل، تبقى العظام هي الأثر الوحيد. من المهم جدا أن يتم تجميع العينات المرجعية من الأهالي ومن من أهالي المفقودين أو من أهالي الضحايا بطريقة مناسبة، سواء يتم تجميع العينات على كروت خاصة تسمى FTA كارد، وهي مميزة لأنها تقوم باستخلاص الحمض النووي مباشرة عليها، وفي ذات الوقت يمكن الاحتفاظ بها في درجة حرارة الغرفة، ولا تحتاج مجمدات ولا تحتاج وجود كهرباء. الخطوة الأخرى هي التعامل بما يعرف بسلسلة الحيازة، بتتبع الخطوات خطوة بخطوة منذ وجود أو إيجاد الجثمان إلى انتشاله، إلى وضعه في المشرحة، إلى أخذ العينة منه وإرسالها إلى المعمل. كل هذه الخطوات يجب أن تتم بتسلسل معين، بحيث لا تتم تداخل خطوة بخطوة. وأيضا الهدف الأساسي هو إعطاء كود معين، يعني رقم مشفر معين لكل جثمان، لكل عينة، حتى تكون النتائج موثوقة ودقيقة في مراحل متقدمة. وجود المعمل داخل الدولة أمر مهم جدا جدا، وخاصة لبناء القدرات داخل الدولة. فمن الممكن إذا تم إرسال العينات دولة ما إلى خارج الدولة، فستكون قواعد البيانات موجودة لدى دولة أخرى، وهذا يشكل خطر نسبي على بيانات المجتمع بصفة خاصة. العنصر الآخر الذي يجب الاهتمام به بعد تجهيز معامل خاصة في الدول التي تعاني من وجود حالات وفيات عالية، وأيضا عدم إمكانية التعرف على هوية هؤلاء الأشخاص، أن يتم تجهيز الكوادر المحلية، الكوادر الوطنية داخل الدولة، لتقوم هي بالعمل، ويكون الإشراف خارجي، ليكن الإشراف من دولة أوروبية أو من دولة أخرى، ولكن يجب أن تكون القدرات التي تبني هذه المعامل وهذه القواعد للبيانات يجب أن تكون قدرات محلية، حتى يتم تطوير الكوادر البشرية الموجودة داخل الدولة. في المجمل، أنا للأمانة حاولت أن ألخص خبرة اكتسبتها خلال السنوات الماضية في ليبيا في موضوع التعرف على المفقودين خلال الصراعات المسلحة التي جرت خلال السنوات الماضية، وأتمنى أن أكون قد أضأت أو ساعدت أو أوصلت معلومة بطريقة ما في موضوع التعرف على الهوية، وخاصة أن الوضع الحالي يستدعي تكاتف كل الجهود لبناء القدرات ووضع خطة واضحة لعملية التعرف على الجثامين مجهولة الهوية. شكرا لكم مجددا.
يعطيك العافية أخت مبروك. إن شاء الله يعني في تعاون. يعني هلا حاليا من خلال المحاضرة طرحنا فكرة إنه إنشاء مركز بحث جنائي أو داتابيس للجينوميك فلسطينيان. فبتمنى إن شاء الله في الأيام القادمة يكون بيننا تعاون لاكتساب الخبرات الكافية.
أتشرف بكم في كل وقت، بارك الله فيك.
خلينا ناخذ بريك قصير لمدة 15 دقيقة، تمام؟ لصلاة العصر اللي عندهم صلاة العصر صارت.